Iron is an essential trace element in all living organisms. It is involved in important biological functions such as oxygen transport and energy conversion processes. In mammals, iron is absorbed in the small intestine by specialized membrane transport proteins. The transport from the intestinal lumen to the cytosol of enterocytes is mediated by the divalent metal transporter DMT. On the other side, ferroportin exports iron, which is subsequently distributed within the whole body. The iron level within the body is tightly regulated, since both iron deficiency and overload has severe consequences. Consequently, the modulation of the iron transporters DMT and ferroportin is a promising therapeutic strategy against iron storage diseases such as hemochromatosis and anemia.
In the iron transporter project of TransCure, groups with expertise in chemistry, structural biology and physiology work together to understand how iron transporters function, how iron transport is regulated in bone development, in the placenta, and how this regulation is disturbed in pregnancy-related disorders. We also try to develop compounds that inhibit iron transporters to prevent the excessive accumulation of iron in the body. Our group is contributing to this work by the detailed structure determination of iron transport proteins and by the characterization of the transport properties.
To study the structural features of these metal transporters, we mainly use X-ray crystallography. That technique involves that we make crystals of the protein we are studying. For that purpose, we first produce the protein in high quantity in cellular systems such as bacteria or mammalian cells. We then purify the protein and concentrate it and then mix it with different cocktails of chemicals to trigger its crystallization. That step is done in this facility, and we use robots to screen thousands of conditions. The crystallization experiments are stored in this device at a constant temperature. Its imaging capabilities allow us to monitor the crystallization process over time. We then harvest the crystals, and their diffraction of X-rays is recorded at synchrotron facilities. From the recorded data, it is possible to obtain the atomic structure of the protein.
Recently, we have determined crystal structures of divalent metal transporters in different states. This has revealed the architecture of DMTs and the way they bind the iron. These structures also show the transition that DMTs undergo during the transport cycle. So first, the iron binds from the outside to a site within the transporter, and then the transporter switches to an inward-facing state where the iron is released to the other side of the membrane. And then the transporter switches back again to an outward-facing state to bind the next iron.
We now can use our structures to understand how inhibitors interact with the protein to interfere with iron uptake. For example, by binding to this aqueous cavity leading to the iron binding site. We can also help chemists to improve the inhibitors to remove unwanted side effects. In that way, we hope that we will be able to contribute to the development of novel therapeutic strategies targeting iron-related disorders.